talking about hemolytic anemia. Hemolytic anemia is anemia which is a reduction in the amount of hemoglobin in the blood due to hemolysis, and hemolysis is the breakdown of red blood cells. Hemolytic anemia can be of different types. What are those types? So the types include intravascular and extravascular hemolysis. So intravascular is simply red blood cell breakdown within the vessel. Extravascular hemolysis is red blood cell breakdown anywhere outside of the blood vessel. So it can occur in the spleen, for example. What is the mechanism of spleen-mediated red blood cell breakdown? That is tagging with autoantibodies of the RBCs. Anyway, what are the other types of hemolysis? There's something called intrinsic and extrinsic hemolysis. What is meant by intrinsic hemolysis? Intrinsic hemolysis is when there's a problem within the red blood cell itself. So some kind of membrane defect, for example, some kind of shape defect, like in hereditary spherocytosis, for example. So that is intrinsic, something which is, the problem lies in the red blood cell, intrinsic. Extrinsic is any problem lying outside the red blood cell. Now that can be anything. That can be literally a spleen problem, a bone marrow problem, anything else. So these are the classifications of hemolytic anemia. It can be intravascular within the blood vessel, vascular problem, or extravascular. What is meant by extravascular problem of the spleen, of the red blood cell breakdown? That means some problem with the spleen, for example. Another way to classify hemolytic anemia is based on whether or not it is inherited or acquired. Inherited can be something like hereditary spherocytosis, and acquired can be something else, okay? So those are the overviews for hemolytic anemia. What is the pathogenesis and what's occurring? So when you have excess breakdown of these red blood cells, remember the normal life cycle of a blood cell is 120 days. Now what we are seeing is blood cells being broken down. When these RBCs get broken down, our bone marrow, which is the central hub for producing all the cell lines, starts to release baby RBCs. Because the mature RBCs are breaking down before their time, they're being hemolyzed. The bone marrow starts to produce baby RBCs known as reticulocytes. So, one of the hallmarks of hemolytic anemia is reticulocytosis. Second, the bone marrow is going to be hyperactive, so you'll get something called bone marrow hyperplasia or erythroid hyperplasia. Third, we can find another characteristic. Now, the bone marrow is not able to meet the oxygen demands by producing enough RBCs, so we have some extramedullary organs. What might be the extramedullary organs that are going to help in the hematopoiesis or the production of RBCs? Those are going to be your spleen. So you'll get extramedullary hematopoiesis from spleen involvement, and the spleen will start to enlarge, leading to splenomegaly. Now, so a clear way of understanding hemolytic anemia, just differentiate what intravascular hemolysis is going to cause and extravascular hemolysis is going to cause. Intravascular hemolysis is basically red blood cell breakdown within the blood vessel. If the RBC is breaking down within the red blood vessel, just imagine a splurge of hemoglobin into the bloodstream. You're releasing a lot of hemoglobin into the bloodstream, which is basically going to turn into heme and globin, right? So you'll have a lot of hemoglobin in the bloodstream. So your first finding with intravascular hemolysis will be what? Hemoglobinemia. Hemoglobin in the blood risen. Hemoglobinemia. Second, you have more hemoglobin in the blood, so you'll renally excrete more hemoglobin, so you'll get hemoglobin urea. Third of all, because you have so much hemoglobin intravascular, remember we're still talking about the intravascular hemolysis, you're going to get this pigment called hemosiderin, and that hemoglobin is going to bind to this pigment called hemosiderin, which is found in our renal tubules. And as our renal cells slough off and start to be replaced and have a turnover, what do we have? We have a pigment called hemosiderin. That's what we use the Prussian blue stain for. And we have excess hemoglobin as well. That excess hemoglobin is going to bind to the hemosiderin, leading to hemosiderin-urea. So now we have 3 characteristic findings of intravascular hemolysis, which include hemoglobinemia, hemoglobin-urea, hemosiderin-urea, and finally you also have hyperbilirubinemia. That's because your hemoglobin is being broken down to hemoglobin, and then you're going to get more and more bilirubin produced, which is going to lead to hyperbilirubinemia, which can deposit in the gallbladder, leading to gallstones. So those 4 or 5 are key characteristics of intravascular hemolysis. Now let's move on to extravascular hemolysis. What are the extravascular organs involved in the production of RBCs? The spleen. So you're going to get splenomegaly. And the spleen and the liver, right? The spleen and the liver are your reticuloendothelial organs. So you will get splenomegaly and jaundice as well. That is extravascular hemolysis.